The development of male gametophytes and pollen formation starts in the flower's anthers in one of 4 microsporangia, or pollen sacs. Each pollen sac holds numerous diploid microsporocytes, or microspore mother cells. Each microsporocyte inside a pollen sac undergoes meiosis to form 4 haploid microspores. And after the microspores undergo mitosis, they form pollen grains, which are the 2-celled male gametophytes. Each pollen grain consists of 1 generative cell and 1 tube cell, surrounded by a tough outer wall. In most flowering plants, the pollen is retained on the anther and released onto pollinating organisms such as bees. The outer walls of pollen grains are very distinctive and are often covered with rough projections that help them attach to the flower's stigma.
Development of female gametophyte. Megaspore mother cell undergoes meiosis to form linear tetrad haploid cell, that is megaspore. Upper 3 megaspore aborts, that is disintegrate, and lowest one towards center of nucellus remains functional. It acts as the first cell of female gametophyte. It undergoes 3 successive free nuclear mitotic division. After first division, 2 nuclei are formed. After second division, 4 nuclei are formed, and ultimately after third division, 8 nuclei are formed. Thus, total 8 nuclei are formed, 4 of which are located at each pole. One nucleus from each pole migrates towards the center and are called polar nuclei. 3 nuclei towards micropylar end constitute egg apparatus. It consists of large central haploid egg cell and 2 supporting haploid synergid cells. Synergids shows hair-like projections called filiform apparatus, which guide the pollen tube towards the egg. Antipodal cells are group of 3 cells present at the chalazal end. The 2 haploid polar nuclei of large central cell fuse to form diploid secondary nucleus or definitive nucleus just prior to fertilization. This 7-celled and 8-nucleated structure is called an embryo sac. This method of embryo sac development from a single megaspore is described as monosporic development. In angiosperm, the development of female gametophyte is endosporic, that is, within the megaspore, female gametophyte, that is embryo sac, is colorless, endosporic, and is concealed in the ovule enclosed by ovary.
To understand the process of fertilization in flowering plants, let us first see all the floral parts. Thalamus or receptacle provides the attachment place for the different floral whorls. Sepals make the outermost whorl. They protect the flower during the bud stage. Petals are generally brightly colored to attract insects for pollination. Anther and filament make the male reproductive part of the flower known as stamen. Stigma, style, and ovary make the female reproductive part called the pistil. In the ovary are ovules. In an ovule is an embryo sac, and in the embryo sac is an egg. During pollination, the pollen grain comes to lie on the stigma. The pollen grain has two layers. The outer layer is known as exine, and it is made up of a substance called sporopollenin. The inner layer is thin and is known as intine. At some places, exine is thin. These areas are known as germ pores, through which will emerge the pollen tube. Pollen grain has two cells. The smaller cell is known as generative cell, which will divide to form two male gametes. The larger cell is known as the tube cell, which will give rise to the pollen tube. The embryo sac has three cells towards chaliza. These cells are known as antipodals. There are two nuclei in the middle called the polar nuclei. Three cells are towards the micropyle. The middle, larger cell is known as the egg. Two smaller cells are called the synergids. The egg and the two synergids would form the egg apparatus. The pollen tube emerges from the germ pore. The haploid generative cell divides to form two male gametes. The pollen tube enters the embryo sac through the micropyle. It enters through the degenerating synergid or between the egg and the synergid. The first male gamete fuses with the egg to form a diploid zygote. This process is known as fertilization. The second male gamete fuses with two polar nuclei to form a triploid endosperm nucleus, which will change into endosperm cell. The fusion of three nuclei is known as triple fusion. Endosperm will divide later on to form nutritive tissue, which will provide nourishment to the developing embryo. The process of fertilization and triple fusion together is called double fertilization.